Our interest is to solve problems in the downstream process of suspension culture of iPS cells. In the upstream stress of suspension culture of iPS cells, cell aggregates are formed and differentiated. In the downstream process, the differentiated cell aggregates must be dispersed into single cells. The problem we are trying to solve is that the process of cell dispersion depends on manual, which makes it difficult to scale up the process. We are trying to solve this problem by developing a scalable device that can be efficiently disperse cell aggregates into single cells instead of manual. To disperse cell aggregate into single cells, shear stress is required to dissociate the adhesion between cells. In the manual cell dispersion process, shear stress is applied by pipetting. The cell dispersion device we have developed is equipped with a simple rotating cylinder, which is characterized by applying a constant shear stress by rotating the cylinder at high speed. If the cylinder rotates too fast, turbulence will occur and damage the cell. And at low speeds, sufficient shear stress will not be obtained for cell dispersion. We designed the device so that the liquid flow becomes a Taylor vortex in the region of optimal rotation speed for cell dispersion. The liquid flow of the cell dispersion device was visualized by PIB, and the shear stress applied to the cell suspension was calculated by imaging analysis. Using a benchtop scale cell dispersion device, a typical Taylor vortex flow was observed at a cylinder speed from 250 to 1,250 RPM. At 750 to 1,000 RPM, which was optimal for the dispersion cell aggregates, the calculated maximum shear stress was 1.1 to 1.6 Pascal. The calculated shear stress was comparable to the value in human capillaries. The characteristics of Taylor vortex flow is that the vortex flow is formed in layers, and it is so that the collision between cell aggregates caused in this state also becomes the device driving force of cell dispersion. The optimal cylinder speed for cell dispersion was compared and verified. As a result, in the cell line used in this study, the yield was maximum at 1,100 RPM, and the yield decreased at higher RPM. It was suggested that there was a cylinder speed that maximized the cell recovery. Next, Oct4 markers were analyzed by FCM to confirm that the dispersed cell remained undifferentiated. As a result, it was confirmed that the undifferentiated state was maintained at any rotation speed of the cylinder as in manual. The cell dispersion reaction solution had comparable recovery efficiencies for manual and device use with triple. On the other hand, PBS containing only EDTA had the same recovery efficiency. In suspension culture of iPS cells, a cell dispersion device was used for passage. Using our bioreactor, cell aggregates grown in 100 milliliter for 4 days were dispersed with a small-scale cell dispersion device to obtain 200 million cells. The obtained single cell was re-inoculated into 500 milliliter cultured for 4 days, and the aggregates were dispersed with a benchtop scale cell dispersion device to obtain 1 billion cells. The obtained cells were inoculated into 5 liter, and 4 days later, 10 billion cells were obtained. For cell dispersion, no enzyme solution was used in all steps, and PBS containing only EDT was used. The obtained cell suspension was not centrifuged, and the dispersed dispersed cell suspension was directly inoculated into the culture medium. The conclusion is as follows. Thank you for watching.